It's really glad to be in Lyon to present this talk. I'm Ning Huang, or you can call me Eric. I'm the first year PhD student at Dr. Sergey Mangu's lab at University of Southern California. Today, I'm going to present you the ongoing project that I'm working on, which is the systematic assessment of the completeness of the TCR sequencing database across diverse mice strains. First of all, just give you a brief background about the adaptive immune receptor repertoires. We all know that the immune responses have been like categorized into like two, which is the first is the innate immunity that is on the left side. The innate immunity just happened like in a relatively short period of time, and it's like work non-specific, just like our epithelial barriers and also like the neutrophil or macrophage. On the other hand, the adaptive immune response or immunities kind of involves in what we know as a B lymphocyte or B cells or T lymphocyte or T cells, and it works more specific, and it works by like the receptors on the B cells or T cell, which you guys heard before, the BCR or TCR that binds to the antigens to trigger the further downstream immune effects. On the genetically structure, the BCR and TCR, they're kind of similar in for BCR, there's like heavy chain, light chain. For TCR, there's alpha chain, beta chain. And how these all came from like the pool of like TCR or V genes, D genes, and J genes, and then undergo what we know as the VDJ recombinations, and like it came with like one unique combination of the VDJ genes and C genes, the constant regions, and to be translated into the TCR or BCR proteins. And among the immunogenomics, one of the most important database in like this field is the International Immunogenetics Information System database, the IMG database. I shows a screenshot of the database here. Also, there's a link to this database, and this database stores the immunogenetics data and also immunoglobulin data, just BCRs and also TCR and also the MHC of human and also non-human like animals, like vertebrate species, and we can just download like the reference of different species of different data, and for like further purposes, like we can do some simulation, and it can be work as some benchmarking purposes. And I do some research about like the number of genes recorded in this database, so we can see like the top three. These are strains. The top three like my strains are have the most V genes here. But our main goal in this project, we just also try to examine how complete is the IMG database in providing us about the mice reference gene. It's because like mice also one of the most important like laboratory animals, and it's relatively cheap, so it kind of use a lot on the in the laboratory sites. And the four mice strain we pick is just like what I just showed. Oh, yeah, this is the four. Not sure if that's the right mice or not, but yeah. And yeah, for the method, it's pretty much straightforward. Like we download the publicly available like TCR sequencing dataset from the sequence read archive, and we run the tool called Mixer. Um, yeah, for Mixer is titled as a universal tool that can help analyze the T and B cells. So it's just a tool that can use in immunogenomics, immune receptor repertoire sequencing analysis. And how Mixer works, just like other like alignment tool, it can help us to build alignments. And what I do is I just specify and align our samples read to the mixer to IMGT reference reads. Okay, so yeah, after running mixer, the third step is just analyze the output from mixer, and we just use this approach. Like the third step, we just use this approach to kind of analyze um the completeness of the IMG database in representing diverse mice strain. Using the first approach is we can calculate the number of mismatches, because our hypothesis that if the reference database is more representing like certain strains, the mismatches in this part, and also the second, the unmapped read, should also be the lowest. So we just have So these are my approach. Like calculate the number of substitution, insertion, and deletion in certain V genes and D genes and J genes alleles, and try to see how it is good at representing diverse mice strains. Yeah, I'm not sure if I. No worries. And these are the samples we have. So we can see there's kind of um, a gap in the number of samples, but I'm still trying to see if I can find more TCR6 samples in like certain mice strains. But these are the current result I have, and it's approximately like 350 or 60-ish samples. So for the result part, so this is the first one. Like I only done the mismatches part, but I'm still working on the unmapped reads part. For the mismatches, we can see like NOD do have the most like statistically significant mismatches compared to the C57BL6J and also the BALB. So we can, based on this result, we can just show like NOD is the less representative in the IMGT database, and yeah, and but the C57BL6 we doesn't expect that this strain gets so many mismatches. So I'm still working on see if what's the issue that caused like C57BL6, which is the second bar, to have that much mismatches. It might be some like quality issues of our samples, but yeah, I will have to investigate that. And yeah, this part, we also should examine the number of mismatches per reads across different strains. And you can see on the legends there, 0 means there is no mismatches per reads. And this plot just shows the percentage of number of the percentage. So 0 means there's no mismatches per read. So it means there's like fully mapped. So it means the IMG database do fully capture the genes from from the 0 from the darker blue ones. And so on the other hand, the 20 plus means there's like 20 plus mismatches per reads. It also means the IMG database are not able to capture that reads, like the genes in that read. And there's possibly will have some part of like nova alleles didn't be recorded in the IMG database reads. And this also a future work I might be able to look at. So look at this paper, list figures, we can just see like NOD is at least 60% incomplete. Why at least 60%? Just like the dark blue ones, it is fully mapped. It's only like above 25%. And the one with like one mismatch, there might be fully mapped, but there's some just random errors in the read. So that's cause there's one mismatches. So yeah, but just based on this, we can also came out with a conclusion like LOD is not that representative in the IMGT database reads. And that's it for my current analysis for the mismatch part. And I'll further still need to work on the unmapped read part. So for the conclusion, what I just said, like TCR database is more representative in representing the C57BL6J strains. On the other hand, the NOD strain is the less representative, and yeah, it's at least 60% incomplete represent in the IMG database reads. Um, okay, so the last point, at least 25% of the reads from the NOD strain are from novel TCR genes, and yeah, that's it. For acknowledgement, thanks like Dr. Sergey Mangu for his guidance. Thanks the lab. Also thanks the previous master students like Yupeng. She helped me to like download and run mixer overload samples. Yeah, that's it. Just I also have a poster session like right at 6 at number 1. You can stop by and then discuss any possibilities, like any thoughts. Yeah. So
Thank you.